invitation. I had always been wanting to be here at the IFT. I think this is the first time I'm talking to physicists. It's quite strange to me. Usually I talk to chemists and material sciences, material engineers. So I'm not used to place many equations in my slides. I'm going to show you more results, more models than the background. So the background, sorry, we can talk, discuss later, but I'm not going to show. I'm going to do as I'm used to. So I'm going to talk about a recent published paper of ours, which shows experimentally the degradation of viruses by nanoparticles using light. So, of course, you are thinking about the SARS-CoV-2. No, sorry, it's a virus of a plant, a virus I have on my orchids back at home, on tomato, on tobacco. But the ideas here are general, and we can apply that for any other kind of viruses and bacteria and other diseases in general. So this is a proof of concept using a specific model, a simple virus that can be manipulated safely in the lab. So what I'm going to show you is related to the targeting of materials to biological systems. When we think about targeting here, we have to think that life on Earth is chiral. Not chirality like physicists, particle physicists like to think. Chirality in chemistry and biochemistry is much simpler. It's related to right hand, left hand. The word chiral, it means hand in Greek. So when Lord Kelvin first proposed the word chirality, he was thinking of that, of two objects which are similar but different. They are equal. If I give you my right hand, you are going to shake it with your right hand. You may try to use your left hand, but it's not going to work. You are going to feel awkward to give the left hand if I give you my right hand. So in biology, this is how it works. So molecules have right hand, either right hand or left hand in nature. In the case of Earth, we have sugars which are right hand and peptides, amino acids which are left hand. This is the basics. So when we think that we are going to target something, we like to think about that. This is the mechanism that we call the lock and key mechanism to have the recognition between different biological molecules. So when we want to develop, I'm going to talk about the nanoparticles, but think about a new drug. If you are going to develop a drug, you have to think about the chirality of that drug. Is the right-hand drug version of the drug be the medicine or the poison? Because it's quite common that one of them works as a medicine and the other is quite toxic. It's a poison to our body. So we have to think that when we go to nanomaterials as well. So if you are going to target biological molecules or biological structures made of biomolecules, they are both chiral, and we have to think about chirality of our materials if you want to have selectivity and efficiency. Both are important. And the problem is that we do not know which enantiomer is going to work beforehand. We have to go to the lab and we have to test both enantiomers. And that's not so simple because materials themselves are not chiral. We have to imprint chirality on them. And this is done experimentally. Most people, at least my age and older, have heard back in the 90s and the 2000s that theoretical chemistry or methods could be used to screen molecules, to screen targets. False. That doesn't work. It's much cheaper, I'm talking about money, it's much cheaper to go to the lab to buy your chemicals from Sigma Aldrich and spend them because simulation is quite expensive. What I'm going to show you would cost easily tens of thousands of dollars of computation. It's much cheaper to go to the lab. So what I'm going to show you is made experimentally, and then I came into play to explain why it works. Before that, I'd like to discuss very briefly that when we talk about nanoparticles, many materials may be used, but usually we are talking about metals and ceramics. Both kinds, the building blocks are simple atoms. You have a kind of nanoparticles made of only metal atoms. Metal atoms are not chiral by themselves, and their crystalline structures are also non-chiral. We may have chiral crystals in nature and in the lab. Yes, we may. The most known is quartz. Crystal quartz of quartz may be chiral, yes, but I'm going to obtain both the left hand and the right hand versions. And that's not good. The racemic version doesn't work. I don't want to have a mixture. I want to have the pure enantiomer, only the single, the left hand or the right hand, and I have to know which one of them is going to work. So I have to imprint chiral information, and the way to do that is to use chiral molecules, which are either of the right or the left hand. There's also, if you have a sense of rotation, like a helical structure I'm going to show you next, they may the helix may grow either clockwise or counterclockwise in the z direction. So these kind of chiral molecules are used to imprint chiral information into the nanoparticles, whether they are made up of metals or ceramics. I also may use a chiral field. I may use a circularly polarized light. The light may be polarized either to the left or to the right, and we can imprint chirality using that kind of light. We are going to begin to study structured light as an option. We think it's going to yield better results, but it's not something I can discuss right now. The paper I'm going to discuss with you has been published maybe 3 months ago. Yes, 3 months ago. And we have proved the concept that nanoparticles have to be chiral if we want them to selectively and efficiently inactivate a virus. So important here, I'd like to show you the names of Professor Nicholas Kotovi from the University of Michigan and Xuanlai Xu from Jiangnan University in China. They are the guys doing the experimental hard work in US and China. And once they have some preliminary results, they come to me and say, okay, we have learned that this material is good for, and for maybe anything in optoelectronics, in detection of molecules, diseases, or treatments, or anything else. In this case, they came up to me before the pandemics. That's the reason I'm not going to talk about SARS-CoV-2, because this investigation began some 5 or 6 years ago. So they tested the particles against a known virus, a plant virus. And after they come to me, usually I have the help of Felipe Mariano Colombari from the CNPEN, the National Laboratory of Biorenewables. And this is what I'm going to show you. But before showing the simulations, the computational simulations, I have to show you the problem, the systems of interest. So this is the virus. You are seeing here 3 microscopies. I have here the bar at 1 micrometer, half a micrometer, 100 nanometers. So you see the virus is very large. Actually, it's not one virus. It's a conglomerate of virus forming a fiber. It's a fiber made up of virus. So this is the typical virus, which is the tobacco mosaic virus. I'm going to call you just TMV. And this virus causes those black dots on the leaves of many plants, including tobacco itself, and any other plant from that family, including potatoes, tomatoes, and so on. So it has some applicability to, as an defensive in agriculture, to reduce the losses in the crops. This virus can lead to losses of about 30%, depending on the culture you are discussing. So this is the virus, and this is the virus after it was treated in the lab with the nanoparticles I'm going to discuss with you, copper sulfide, the nanoparticles, and you can see that the viruses are no longer there. They have been dismantled. So what is the results that have been gathered over the years? I'm going to call for now, before I show you the actual nanoparticles, the G-type DNPs, the L-type LNPs, and the REC-type, the racemic, the mixture of both handedness. And you can see the efficiency with which the nanoparticles can inactivate the virus, and the light that has been used, the green light at 532 nanometers, and the light might be right circularly polarized, linearly polarized, left circularly polarized, or a combination of both. So you see that the D nanoparticle interacting with the light polarized to the right yields a very high efficiency. If I pick up the same nanoparticle but use linearly polarized light, efficiency decreases by 1/3 to 1/3. And if I change it to the light polarized to the left, efficiency is 0. So the same nanoparticle is activated by one-handedness of the light. It loses its activity if you use a light without circular polarization. And if you invert, if you switch the polarization to the left, it goes to 0. If you pick up the left-handed nanoparticle, the results are similar, but you see again that there are situations when you have 0 efficiency. In particular, the racemic nanoparticle has a very poor result here with any kind of light. So the chirality of the nanoparticle is the most important factor here. And you see that you have something different, because when you go to the left nanoparticles with the lefty light, the efficiency is not comparable, it's slightly lower. So you have two things here. You have, it's indicating that you have a larger thermodynamic preference for the D type with respect to the L type. And you also see that you have some difference in efficiency depending on the polarization state of light you are irradiating the sample. So those are the questions that have been posed to me and my co-workers. And we have to model the virus. But the virus is very large. I showed you the microscopy, the virus with hundreds of nanometers in length. But when we go to the database of structures, you find that you do not have to consider hundreds of nanometers. A few nanometers, tens of nanometers should be enough. This structure is deposited. It has been resolved for the fiber, like those I have showed you. And I have here 49 proteins forming the capsid. The same protein packs together in a helical structure, and it can grow longer and longer, but this is the smaller size which can fit the RNA of the virus inside the capsid. So this is the smaller model I can handle to describe this virus, and it's already a very large model. I have 133,000 atoms here, just the protein. And I have to place the protein inside water. I have to place my nanoparticle and so on. So you're going to, you can already imagine that the model system is going to be very large. This number of protein units, 49 protein units, is just enough to make 3 complete pitches of the structure, and it's stable. If I place this alone in water and run a molecular dynamic simulations, the structure is very stable. So this is the model we are going to discuss. The nanoparticle, this is from the paper. I have a problem here. Most of you are physicists, of course, but when we see a non-integral number here, it means that my structure is very complex. We call it in chemistry a non-stoichiometric compound. It means in practice that the unit cell of these ceramics is very large in size and in number of atoms, and it means that I have different types of copper atoms and different types of sulfur atoms inside that unit cell. Chemically, they are different. So it's very challenging to make the computer simulation of that kind of system. Experimentally, it's very simple to be made, not complicated. And when you finish the synthesis, this is what we're going to see. Nearly spherical, not perfectly spherical, nanoparticles. The size bar here is about 2 nanometers. So what we are seeing here is a nanoparticle in the size range of 2 to 4 nanometers. There's a size distribution. You may have sizes as small as 1 and as large as 10, but the most common size here is 3 nanometer. So this is the size we are going to work in our computer simulation. But how can we do that? Oh, just one information here. When we take these pictures from the transmission microscope, we like to make these measures, and we say, okay, this is the phase 200, 311. So these are the crystalline phases, the Miller indices of the crystalline phases we have to use to draw our nanoparticle. So what we need the structure of the crystal, and we need to cut to make our model it should have the same size and the same shape of the actual nanoparticle. So this is our nanoparticle. In that microscopy, you cannot see the organic molecules. They are there. The molecules, in the previous slide, they are drawn here, is penicillamine. So this penicillamine here, it's an amino acid, and this amino acid is chiral. It's chiral because it has a carbon atom here, the alpha carbon atom, which is connected to 4 different ligands. So this makes that carbon atom chiral. And that's the molecule that's going to imprint chirality here. So when I say that the atoms are different, it means that I have copper atoms bonded to copper atoms, bonded to sulfur atom to sulfur atoms, and sulfur atoms to copper atoms, and so on. The coordination of the atoms changes a lot. So it's a very complicated system to study. So we have made our model drawing a supercell of the chirality, and this structure was cut using those crystallographic information from the microscopy. And we have made some DFT calculations using the semi-empirical Hamiltonian GFN1XTB. It's a very, it's recent Hamiltonian, and it's very efficient and very accurate. It's not accurate, for instance, to give you the heat of formation. It yields bad results for heat of formation, but it's very nice for the structure, the atomic structure, the crystalline structure, and also for the electronic structure, meaning that it yields very nice spectra for the electronic excitations. So the electronic excitations were made using this Hamiltonian with this simplified Tandenkopf approach. The Tandenkopf itself is approximation. This is the approximation of the approximation because I have to deal with thousands of atoms, tens of thousands of electrons. So it's very huge. I cannot perform a better calculation here. Okay, so this is the model system. And how can I say that it works? I do not have anything else to compare except the experimental result. And the best result I have is the dichroism, the circular dichroism, which can be calculated and compared with the experiment. So it may seem not too similar, but it's a perfect match in the area of material science. Because what we have to look at the not the exact position, but the exact sequence of negative and positive bands and the relative intensity they have. So I see here that we have, for instance, for the D, which is here, the colors are not very nice, but okay. The red with the violet here. We have a shallow and broad band above 700 nanometers. Okay, I have it here. I have a higher intensity peak, a negative peak closer to 600. It's shifted to lower wavelengths, but it's okay. I have a bumping here. So I have the same features. So this is the best match you may expect from a model system in nanoparticles calculations. So we consider the model nanoparticle is good enough. So this is the actual simulation we performed, the molecular dynamic simulations. I'm not showing the water molecules because you would not see anything. But you can see the virus particle here. And we placed 7 nanoparticles inside the box. And after 350 nanoseconds, one of the nanoparticles entered into the pore. And that's important because experimentally it has been determined that the virus is inactivated because one single chemical bond of the protein is cut and that bond is inside the pore. So the nanoparticle has to be there. Why I say that nanoparticle has to be there? Because experimentally it has also been determined that the mechanism involves the so-called reactive oxygen species. A free radical. A free radical is not selective. It eats anything on the way. So if it cuts the same bond, the chemical bond, for all the proteins, for all the viruses, it has to be generated in situ. It has to be generated inside the pore and very close to the position where the reaction is supposed to occur. So that nanoparticle is our focus now. We are looking at that nanoparticle. It's a very large simulation. We have over 1 million atoms in the model system. But we know that we have to look not at this one or this particle or these, the other 6 particles. They absorbed on the outer surface of the capsid. But the only particle inside the pore, that's the particle we are interested in. So we are going to look at that particle. So here I have the simulation that was for one of the systems. The same simulation was run for G nanoparticles and L nanoparticles. You have different dichroisms calculated there. Those dichroism curves are average over 10 structures. And the model system is a piece of the actual system. We pick up that nanoparticle and the proteins around, the proteins in contact, direct contact with the nanoparticle, and the water molecules and the counter ions and everything else that was close enough. So what we are seeing here is a piece of the model with about 5,000 atoms. And that's the 2 systems that we use to make the quantum chemical calculations. I'm going to run the video now. What we're going to see here is the best that we can expect to do with the electronic excited states, the excited electronic states of the system. The number of excited states in that spectral window is in the order of 10,000. You cannot expect to look at each one of them to see, oh, I'd like to see the excited state number. It makes no sense. So what's this video? I'm going to play again. Let me just explain. We averaged the states in a window of 10 nanometers of wavelength. And for those excited states, we computed the change in the population. You can use Millikan, Ludwig. You use a population, and that population, the difference between the ground state and the excited states tells you about the electronic polarization. So what we are going to see here, I'm going to play again. When you see red isosurface, it's the region where the electrostatic potential became negative. A blue surface, it's where it became positive. And that's important because the polarization produces the radical species, the reactive oxygen species. So we would like to see curves where the polarization goes closer to the protein fragments because the excitation has to occur very close to where the reaction is going to occur. I cannot form the radical species, the free radical, far away because it's going to react with something else which is not my target. And that's not observed experimentally. So, playing again. Now you know what we are looking at. The isosurfaces depict isosurfaces for more or less 5 millivolts. This is the region 534 where the, I can play again and stop at the position I would like to show you. Where the experiment is made, we're using green light. So closer here. Oops, a little bit before. No, I'm not good with that. Again. When you do not see a surface, it doesn't mean that nothing happened. It means that the ISO value was not so large. Okay, I guess yes, this exact position. So you see that when you see the L enantiomer, these molecules down here are the protein fragments. You do not see any polarization whatsoever taking place where the proteins are located. So this is why this kind of enantiomer is not so efficient. And on the other hand, the polarization for the D particle takes place exactly where the proteins are located, on the right of the nanoparticle. So the location is correct. But this is too large, so we refine it a little bit. I have to play again before moving on. Okay. We did a more detailed calculation. Okay. Just for the single protein which was closest to the nanoparticle. We cut the extra atoms and reduced the size from 5,000 atoms to 1,000 atoms only. So now we have the whole nanoparticle, and we have the single amino acid, and the chemical bond that breaks is depicted here with this surface, and here with this surface. What you see here, you see that the chemical bond red means that it has lost electronic density, while this nearby water molecule has gained electronic density. So we have an evidence here that, yes, the chemical bond here is weakened by the excitation, the electronic excitation, and a nearby water molecule is probably a candidate to become a radical species, a reactive oxygen species, taking the electron to itself. While the L enantiomer, you cannot see any relevant electronic polarization whatsoever for this protein adsorbed to the surface. So there are many other results, but these are the main findings from the simulations that show us that the probable mechanism of action of this nanoparticle. So how about SARS-CoV-2? I may just give you a teaser. Experimentally, my co-workers have synthesized the nanoparticle with this tear shape. And it's interesting because the pointing ending interacts with the proteins of the virus. The spike proteins, they form these conglomerates. And if you have D particles, it's not so nice. But when you have here the L particles, see, the other enantiomer is more effective here. They seem to pinpoint the spike. And the computer simulations show the same features. The L enantiomer, the point of the nanoparticle goes straight to the RBD, to the position in the spike protein where it's supposed to interact, while the D enantiomer has a not so interesting interaction, a lateral interaction, and the racemic nanoparticle as well. So this is just a teaser, paper submitted 3 times in 2 weeks. It means it has been rejected 2 times in 2 weeks. Fast rejection. So I have to acknowledge all the fundings we have had, especially the supercomputer Santos Dumont. We could not have done that without them. And thank you for your attention.
So very, very interesting talk. I have a question regarding the activation as a function of the wavelength of the laser, that you showed that for specific wavelengths, the activation is actually larger, and there's some regions. But then in the experimental table, I saw the last 2 lines were experiments done with sunlight, like simulated sunlight, yeah. And the efficiency is above 90% on those, yeah. 2. So. Yeah. So does the wavelength actually matters, or if you just shine?
Yeah, it matters. It matters when you ask why that a specific wavelength, because the laser has that specific wavelength. So you are going to use commercial lasers, so you are going to use the commercial colors you have. When you go to the actual application, the problem with natural sunlight is that you cannot control the power over the day and the year and the season, so you make that artificial illumination mimicking. So yes, you have seen the full spectrum. You have many interesting bands there. So if you are going to use just one band, you are focusing on one band, but it does not mean that other bands are not important.
But if you shine sunlight, you know...
It works. It works. So this is the good news. You do not have to put polarizers on the crops. That's the point.
Okay, I have the same question about polarization of sunlight. So.
Yep.
But what about polarization in your light polarization in your simulations? How does it enter in
the calculation? It does not.
No, in the okay, in the reaction. Okay, it doesn't enter. Okay.
Yeah, the polarization in the quantum chemical calculation. Is that your question?
I mean, in the model, how can you see in your model that the right polarized light works better than
okay.
polarized?
No, we cannot say that because the quantum chemical calculation does not take into account actual light. It's a perturbation procedure to compute the excited states, and you can obtain the spectra and say, okay, if it's positive, it absorbs more the L, the left circular, than the right circularly polarized light. But it's a fact. It's not something we place in the model. The light's not there. The sign of the band tells us which kind of polarization interacts better. The orbitals are the same, but the efficiency of absorption changes. This kind of material, the difference between absorption between the left and the right forms of light, it's very small, 1% at most. But depending on the system, you can have a system that absorbs one handedness and nothing of the other. That's possible, but not necessary. The results tells us that we should not pursue that. It's hard to make, and it's not necessary to have the absorption of a single-handedness without the absorption of the other. We may have the absorption of both. If we have an excess of one, it's enough.
So, yeah. Very nice work. So, just for me to understand, the nanoparticle itself is not chiral.
No,
so
it's not chiral.
What is chiral are the molecules that are attached to the nanoparticle.
Not completely true. If I pick up the nanoparticle before placing the organic molecules and perform the calculation, I'm going to see bands, circular dichorism bands there. But both handedness are possible. So in practice, the nanoparticle in the real world would be racemic. So the mixture of both would yield zero dichroism.
So each nanoparticle is both.
Yeah.
It's not one
if you
nanoparticle.
think about water, liquid water, if you pick up a cluster of water from your simulation and perform a calculation like this, you are going to see that your cluster is chiral. But on average, water is achiral.
Okay, now my question. So what is the role of the nanoparticle then in the whole process? Why can't you put just the chiral molecules there and try to get a similar...
if you go to the support information, you're going to see that placing the organic molecules there yields zero result. So you have to have the nanoparticle for many reasons. In this case, copper. Copper atoms are known to be toxic to cells, to viruses. You use Bordelaise tincture. It's a mixture of copper sulfate. Copper is known to be active because it produces free radicals. Copper is necessary.
Okay, so let's finish the session and thank the three speakers again.
Guys, we're going to take the group photo. So if you want just to step outside, we'll take the photo here before coffee break.